So this is part 4 of chapter 13, and we're talking about crossing over. So a couple more things to point out about the crossing over or exchanging of DNA between homologous pairs is that the sites of where this crossover occurs is called a chiasmata. And so the chiasmata are simply where these sites of crossover occur. And again, I'm not sure that if I got cut off or not at the end of the last video, but I was saying that they're researching this a lot to figure out how we get exact lining up of the genes, because they have found that there's not a loss of genetic information through these crossovers, that genes remain intact. So in some way, this process is occurring to enable exact switches between sets of data here. And often the average number of crossovers that occur per tetrad or synaptonymal complex is between 2 and 3. So it's not just one crossover that will occur. There are between 2 and 3 on the average that will occur for every meiotic division. Okay, so you can see here, this graphic here is showing the cohesins that are holding the sister chromatids together. So that has to loosen up, and this here is where we have the chiasmata, that crossing over portion. And with that exchange of this portion moving over here and this portion, well, actually it's already occurred here. So this is the one chromatid that's taking over this portion, and this chromatid's taking over this portion, so that when we have anaphase 1, we have the separation of these sister chromatids, or this homologous pairs being separated here, taking with them the new DNA on only one of the chromatids. So then when we go through mitosis 2 and we have anaphase 2 occurring, you can see here how we have the same sister chromatids here, but they're not the same. So one of our daughter cells is going to end up with one chromatid that was just like the original that we started with from the mother. But the other chromatid is actually going to carry some of the paternal DNA with it. So we end up with a different combination of genes here on this chromatid. So this is how we're going to be getting that variation in genome that we're looking for. So we will put up with our different sounds there. So again, looking at how we were needing to get gametes that were haploid, we are seeing the same slide here where we have meiosis going from diploid to haploid to get these gametes, and then these haploid gametes fuse to return to a diploid zygote. And then the zygote is going to go through just regular mitosis, where the original zygote cell that is diploid is going to make identical daughter cells, and as the zygote grows into our embryo. Okay, these are just some photographs to show you what real slides or real cells would look like during the different phases of meiosis. So you can see here in the prophase 1 where we have our sister chromatids, and you can see right here that we have a homologous pair right there. Here's metaphase 1, anaphase 1, metaphase 2, anaphase 2, and telophase 2. So it might be good if you can pause this and just study these and see if you can point out some of the differences and talk through some of the differences of these different stages of meiosis. So to recap all of this, we're going to look at a quick animation going through the different steps of meiosis.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
So the last thing that we want to focus on here is how sexual reproduction has the benefit of introducing genetic variation, and it does it in 3 ways. We have, first of all, independent assortment of chromosomes, which is essentially the random alignment of the homologous chromosomes during metaphase 1. We have crossing over, which occurs during prophase 1. And then at the very end of our process with our gametes, we have random fertilization. So the different gametes, like different sperm, will fertilize different eggs, and that combination will end up giving a different variety of results in the offspring. So here we'll look at this. Here we have 2 equally probable arrangements of chromosomes at metaphase 1. So looking at what is independent assortment. So here we have, what is this cell in terms of its diploid number? It's a 2n equal to, you should say 4. So we have 2 chromosomes from the mother and 2 from the father. And how they line up during metaphase is a random thing. It's an independent assortment. So in this case, we have these two lined up the same, but the bottom two are lined up opposite. And so how they line up is going to determine where they're going to end up in which daughter cell. So that when we come down to metaphase 2, how we have these dividing up here during metaphase 1 is going to end up giving us completely different daughter cells at the end of meiosis 1. So you can see here these two ended up in one daughter cell, and these two ended up in this daughter cell. Where here we have these two ending up in one and these two ending up in the other. And then as we continue through meiosis, you can see how we end up with totally different gametes. So this is how we get variety through independent assortment. We also get variety from the crossing over events. So crossing over is going to create completely new combinations of traits on each recombinant chromosome. So that's a term that you should be familiar with. When crossing over has occurred, what we end up with is a recombinant chromosome. Because that makes sense. We have a chromosome that has recombined genetic information. And again, in humans, an average of 1 to 3 crossover events occur on every chromosome. And then lastly, we have variation from random fertilization, where we have the sperm and the egg equal our question mark. And so you can see here with our pictures, well, this actually is kind of a more interesting picture. This larger sphere is an egg, and these are all sperm. So which sperm is going to end up with this particular egg is a random event. And the possible combinations from independent assortment gave us 2 to the n, where n is our haploid number. So if through independent assortment we end up with 2 to the n possible combinations, so with humans our haploid number is 23, so 2 to the 23 is how many possibilities we can end up with in our individual gamete. So our individual sperm or our individual egg that has resulted from meiosis. So if we have two parents, we actually end up saying we have 2 to the 23 possibilities times the second parent, 2 to the 23 possibilities. So that means we actually end up with 70 trillion possible diploid combinations in our zygote. It's pretty mind-boggling, and it actually explains how people end up looking so unique and have such unique traits. So this is showing us, here's one couple with totally different offspring, and here's another couple with very different offspring. Sexual reproduction allows us to maintain both genetic similarity, but there's also all the differences, and these are just some fun celebrity families. So you can see the Kardashians, how there's some similarity, certainly in hair color, but a lot of variety as well. Same thing with the Jonas Brothers. Pretty similar hair coloring, just general skin coloring, but they look different. And then these Baldwin brothers, you can see similarities and differences. So this is our final animation here, but I'm actually running out of space on this video, so let's see, we'll just go ahead a little bit here. Just in terms of spermatogenesis, how sperm are produced, it's through meiosis, and it's very straightforward. So everything that we've been learning so far about meiosis is how we end up with our 4 gametes at the end of meiosis 2, which develop into sperm. So every time we go there's meiosis, we're going to end up with 4 viable sperm that are